Hello students. Students, in this session we are starting with experiment number 3 of your biology practicals, and the name of this experiment is study of plant population density by quadrat method. Now students, previously we have already done two of the experiments. The first experiment, we did the dissection of the hibiscus flower where we studied the floral whorls, and in the second experiment we did the process of isolation and extraction of DNA. Okay. Now in this experiment, we are going to study plant population density. That means how to take out a plant population density, or you can say how to study the plant population density using a quadrat. So let us just quickly start with this experiment. Now the aim of the experiment is to determine population density of plants by quadrat method. Now students, when you speak about population density is defined as the number of individuals of any particular species in any unit area. Okay? And when we are studying about the population density of plants, that means the numerical strength of a particular plant species in any unit area. Okay? And to determine this population density of that particular plant species, we are using this quadrat method. Okay? So students, this is the quadrat, the diagram of the quadrat, and this is the actual quadrat that is used in the fields. Now saying this, this actually the experiment that we are doing today is an experiment that is done in the field. It is more of a field work over here, okay, where you make such quadrats and then you determine the population density of the plant species. Now mostly for herbaceous plants, studying the herbaceous plants in the field, the quadrat that is made is 1 meter square quadrat. And this 1 meter square quadrat is having these 10 centimeters coordinates. Okay. Now if you want to make this quadrat in the field, when we are studying about the plant population density in the field and we are using this quadrat, there are certain requirements to make these quadrats. You need a meter scale to measure the quadrat. You need the string and the cord to make the boundary of the quadrat. Then nails. Now why do you need nails? So that you can fix these nails at 4 corners and you can make a boundary out of it. And then you need paper and pencil to calculate the population density of the plant. Okay. So let us just quickly see the principle of this experiment. Now the principle of this experiment says that all the plants of a species growing in a geographical area at a particular time forms plant population of that area. Okay. So what is a plant population of a particular area? The plant species growing in any geographical area at a particular time. It forms the plant population of that area. Now this plant population of a particular species, it actually changes from time to time. Now when we say that it changes from time to time, it may increase or it may decrease. That means the population of that particular species can increase or it can decrease. And then this increasing and decreasing of the population of the plant species, it depends on various factors. Now these factors can be abiotic factors or it can be biotic factors. Now when we speak about biotic factors, it can be various bacteria, various insect pests or various viruses that are controlling the population of that particular species in a geographical area. And when we speak about abiotic factors, it can be like the salinity of the soil, or it can be the metal toxicity, or any climatic conditions like too much of rains, flooding you can say, or famine. Any such abiotic factor is also responsible for changing the population of that particular species in an area. Now, the number of species present per unit area at a given time is called population density, is what we have already discussed previously as well. And this population density, it can be calculated by this formula. Okay. So D stands for the population density, which is equals to N upon S. N is the number of individuals of that particular species in that area. Okay. So whatever is the number of individuals of a particular species in an area is denoted as N. And S is the unit of space of area. That means the number of quadrants. So if you're studying one quadrant, it will be 1. If you're studying 2 quadrants, 2, and that is how it goes on. So that is how population density, it can be determined by marking the quadrants of suitable size and recording the number of individuals of each species occurring in that quadrant. So as I told you, mostly for when you're studying about the herbaceous plant, the quadrant that is made is of 1 meter square. And depending upon the size and the frequency of the plants that you're studying, the quadrant can change. The size of the quadrant can change accordingly. So this is the principle of this experiment. Now let us quickly get on to the procedure. So to start this particular experiment, now again let me tell you that this experiment is a field work. Okay, so this procedure is according to the work that you actually do in the field. So to start this experiment, you have to select a field and the plant species to be studied in that field. Okay, so you have to select a field and then you should also know the species that are growing in that field so that you can select certain amount of species for your study. After that, after you have selected the field and the plant species, then you will fix 4 nails A, B, C and D at a distance of 1 meter, making a square area of 1 meter square. So if you can see over here in this picture, there are many quadrants. There are about 10 quadrants that are made, and each quadrant is made by fixing these 4 nails A, B, C and D, which is at a distance of 1 meter, making a square area of 1 meter square. So after fixing the nails, you have to tie a string around these nails so that you can make a boundary, and this makes 1 quadrant. Now again, the same thing, that the size of the quadrant, it should be determined on the basis of the size and the frequency of the plant species that are growing over there. Okay, and that plant species should be the species that you're studying. Okay, now next that you do is the count the number of plants under study lying inside the quadrant. Now if you can see in this picture over here, this plant with yellow flowers, this is marked. So suppose this plant with yellow flower is the plant species for study. Okay, so you will count the number, the particular individual of that species in each quadrant. So if you see in this first quadrant there are 2. In the second quadrant there are none. Even in the third quadrant there is no such plant species. In the fourth quadrant they have marked 1, 2, 3, 4 and 5. So there are 5 of this plant species in the fourth quadrant. Fifth, there are no such plant species. Sixth, there are again 1, 2, 3, 4, 5, 6, 7. Then same thing goes on with all the quadrants. After this, you have to record the data in the observation table. That means in the first quadrant, how many did you count? Second quadrant, how many did you count? And so on for all the quadrants. Once you record the data, then you have to calculate the population density using this formula. Okay, so the formula is population density, that is D is equals to N upon S. So N over here is total number of individuals in all the quadrant studied. So suppose I do a random calculation of about, you can say there are 10 species in all the quadrants. So here there will be 10, and how many species are you studying? How many quadrants have you studied? There are 10 quadrants that you have studied. So 10 upon 10. Okay. This gives you the population density of that particular species. Understood? So students, now this particular experiment, as I told you, it is a field work. Now here we are doing it in the lab. So I have made certain arrangements to study this particular experiment in the lab. Now what I have done is I have taken this tray. So suppose this is an artificial field that I have made, and I have divided this artificial field into 4 quadrants. So this is quadrant number 1, quadrant number 2, 3 and 4. Okay, so we will be studying 4 quadrants, and these are the species of the plants that we have taken for study. So the first species is Duranta speciosa. Okay, the second one is Citrus lemon. The third one is Zapyranthus, which is a type of a lily. And the fourth species is the random weed that grows along the roadside or in the field, which is Amaranthus species. So we are going to study these 4 species in these 4 quadrants. Okay. So let us start with this experiment. Let us start studying about these species and let us record the data, and then we will calculate the population density of each of this species into these quadrants. So I have already made this observation table where I have already written the data, and I have also done the calculation of the first species for you all to understand it. Now let us see the rows and columns of this observation table. So first thing is the serial number. So 1, 2, 3, 4 serial numbers. So that why? So that we are studying 4 species of the plants. And the plant species are again species number 2, that is Duranta. Species number 1, yeah, that is Duranta. Species number 2, which is Citrus lemon. Species number 3, which is Zapyranthus, and species number 4, which is Amaranthus. Number of individuals per quadrant. So there are 1, 2, 3 and 4 quadrants, as you can see here as well. And you have to record each species in each quadrant. Then total number of individuals in all the quadrants. Okay, that becomes your n. So if you see species number 1, there were no such species in quadrant 1. So as you can see, the first species is Duranta. So this is the Duranta plant for your information in this quadrants. So if you see in the first quadrant, there is no Duranta. Okay, so I have put no value over here. In the second quadrant, there are 2 Duranta species, so I have put 2. In the third quadrant, again you can see no Duranta growing. So there is again no value in the third quadrant. And in the fourth one, there is 1 growing, which is over here. So that is 1. And if you total it, it will become 3. So total number of individuals in all the quadrants studied is 3. Then total number of quadrants in which the species have occurred. So out of 4 quadrants, the species have occurred in 2 quadrants. This is 1 and this is 1, the second and the fourth quadrant. So here the value will come as 2. Then total number of quadrants that you have studied. How many quadrants have you studied? 4. So out of 4 quadrants, this particular species has occurred in 2 quadrants. So I have studied 4 quadrants. And then you take out the population density as D is equals to N upon B. Now students, here I have taken B, you can take it as S. In the observation table that was given in your handbook, the alphabet that was denoted was B. So I have taken over here as B. So this is 3 upon 4. Okay, 3 is total number of individuals in the quadrants, and then B is total number of quadrants that you have studied. So if you calculate this, the value comes as 0.75. So 0.75 is the population density of Duranta speciosa. Okay. Now next, species number 2, which is Citrus lemon. Let us calculate the population density for this particular species. Okay. Now, if you see in the first quadrant, species number 2, that is citrus lemon, has occurred once. So this is 1. In the second quadrant, we can see again that this particular species of citrus lemon has occurred once more over here. So in the second column also you will put 1. In the third one, that is this quadrant, it has occurred twice, 1 and 2. So in the third quadrant, in the third column, I'll put 2. And in the fourth quadrant, there is no such species, so I'll put a dash. Okay. Then total number of individuals in all the quadrants studied. So 1 plus 1 plus 2, that goes for 4. Okay. Total number of quadrants in which the species has occurred. So out of the 4 quadrants, the species has occurred in 3 quadrants, that is 1, 2 and 3. So the value that will come over here is 3. Total number of quadrants that you have studied, so you have studied 4 quadrants. So again, the value that will come over here is 4. And then you have to take out the population density by D is equals to N upon B. So your N over here is 4 and your B is again 4. So 4 by 4 is equals to 1. So the population density of citrus lemon is 1. Similarly, we have to find it for species number 3. So let us now do for species number 3. Species number 3 is Zapyranthus. Okay. So Zapyranthus is this that is growing. Okay. It is a type of a lily. So in the first quadrant, that is this, there is one Zapyranthus growing. So again 1. In the second quadrant, there are no Zapyranthus growing, so dash. Then in the third quadrant, there is again 1 growing, so 1. In the fourth quadrant, there are 2 that are growing, so 2. Total number of individuals in all the quadrants, 1 plus 1 plus 2. That goes for again 4. Total number of quadrants in which the species has occurred. So the species has again occurred in the 3 quadrants out of 4. So 3, and number of quadrants that we have studied is 4. D is equals to N by B. So N is again 4 by 4. So that again goes to 1. So again the population density of Zapyranthus is 1. Now the fourth and the last species, that is Amaranthus. Now Amaranthus, in the first quadrant, it has grown once. This is Amaranthus, so once. You can see no Amaranthus growing in the second quadrant, so dash. In the third and the fourth quadrant as well, there are no Amaranthus growing, so again I'll put a dash over here. So total number of individuals in all the quadrants that we have studied is only 1. Total number of quadrants in which the species has occurred is again 1. And total number of quadrants that you have studied remains 4. And then if you do 1 by 4, the value that you get is 0.25. Okay. So that is how, students, you have to take out the population density of all the species that you are studying in this field. Now students, let me tell you one thing. Please do not enter these values that I have told you all right now in your journal. The values that you have to enter in your journal, it will be given separately. Okay. So this is just for your understanding. Please do not enter these values in your journal. Okay. Now this was the observation. We have recorded the observation, and on basis of this observation, we'll determine the result of this experiment. So the population density of the given plant species, species 1 was 0.75. So 0.75. 0.75. Species number 2 was 1. Species number 3 was again 1. And species number 4 was 0.25. And then let us just conclude this experiment. So for conclusion, the population density of the highest and the population density of the lowest species we have to determine. So highest out of these is species number 2 and species number 3. That was citrus lemon and Zapyranthus. So here you will write citrus lemon and Zapyranthus, and lowest was for Amaranthus. So species number 4, that is Amaranthus. Okay, so that is how the density value is expressed as the number of individuals per unit area. So students, with this we complete this experiment. In the next session we will be starting with one more practical, where we will be doing the 4th experiment of your practical.